So that the concept of exhaustion is very important. I want you guys to be very clear on this thing, that how, and you can figure this thing out as well, that a normal cell, when it becomes cancerous cell, it does not change everything. It actually, it changes the most of the receptors present on the cellular surface, they stay same. Most of the DNA stays same. Most of the chemicals the cell is producing is same. And using those receptors, which are like which is like a marker of a normal cell, a bad cell can avoid the immune system. Actually, it's a kind of cheat. They can cheat the immune system. And here, two of them are mentioned. The one is the PD1 and PDL, and the other is the CTLA4. We have talked about the CTLA4 earlier, that how it actually block the activation of the T cells. But here, the PD1, PDL, L means the ligand. One of them is the ligand. PD1 is the receptor which receives that ligand. Okay. Other is this combination. So scientists has developed a drug. They thought that if we, for example, this is the this is the bad cell, or I must say the cancer cell. It has other things, but it also contains the receptor called PD1. So that means how, no matter how much bad is this cell will be, our immune system will not going to kill that cell. Why our immune system will not going to kill that cell? Because our immune cells carries the PD1L receptor, the ligand receptor of that one. Once that combination is established, it means this is our own cell. We don't need to kill it, and that cell will survive. This is the reason that most of the cancer cells in the early stage, they manage to fool or cheat. They manage to avoid the immune system until unless they started producing something very weird, or they started having a very different receptors out there. So scientists has developed a drug, a molecule. That molecule binds with the PD1 or with the PDL. That drug binds with the PD1. So in that case, what will happen? If the drug bind, for example, this is the CTL. आपने मुझसे सवाल पूछना है इसके ऊपर, ठीक है? तो शाबाश होएगी. They create a molecule, a drug molecule, which binds with this PDL1 ligand, which actually blocks this one. If this one is blocked, that no combination is possible here. That means that CTL will now get the license to kill that cell. So the bad cells, which were, or the tumor cells, which are impossible to kill because of the presence of this PD1 protein on their cell surface, are now prone for this process, are now visible to our immune system, are now, our immune system is able to kill those cells as well. Okay? So that's how people are trying to cure the tumor cells. So in the tumor, all these cells have the PD1 domains. So all of them are safe. This CTL is not able to kill any of them, although all of them are bad, because all of them are carrying the very famous PD1 receptor. So now what you are doing, you are actually blocking the PD1 receptor present on the CTL. If you block this receptor on the CTL,
sir.
Shabash.
Sir, how does that drug only attach with the tumor cells and not the PD1 of a normal cell?
Okay, so I told you you should ask me a question. I will give you a shabash, so you will get the shabash. Okay, you just got it. शाबाश, मैंने आपका नाम लिख दिया इसलिए आप CTLs जो होते हैं ये, यहाँ पर दो क्वेश्चंस आप मेरे से पूछ सकते थे. One of the question is that, sir, you block the PD1 domain on CTLs. So how it is possible कि सिर्फ
ये सर दूसरा क्वेश्चन मेरा था,
सर
ये नॉर्मल सेल्स को भी तो फिर किल कर सकता है
शाबाश.
वहाँ पे PD1 के साथ अटैच हो जाए ड्रग.
ओके राबिया, शाबाश, गुड. द सेकंड क्वेश्चन इज दैट ये कैंसर सेल्स को किल करेगा, तो ये जो प्यारे प्यारे से जो है नॉर्मल सेल्स रह रहे हैं वहां पे इर्द गिर्द, इट विल स्टार्ट किलिंग देम एज वेल. यानी कि इट विल गोइंग टू बी, क्या वर्ड इस्तेमाल करूं इसके लिए, इट विल गोइंग टू बी एन इनडिस्क्रिमिनेट किलर. एम आई राइट? It will going to be an indiscriminate. It will not be able to discriminate between our own cells and the cancer cells. It will start killing the both. That's very bad. So how the scientist thought about it? उन्होंने सोचा कि यार ये कैसे इसको, जैसे आप लोगों ने क्वेश्चन किया ना कि if we block the PD1 domain on the CD8 cells, they will become indiscriminate killers. They will not only kill the bad cells, but they're also going to kill the good cells as well. ये कीमोथेरेपी में होता है ऐसे, if you remember, they kill the good and bad cells together. ये बड़ी molecular level की कीमोथेरेपी है. I wish you guys are with me on these things. So what they are doing is that they create those cells, or they give that drug. They give that drug with the help of an injection. एक injection लगाते हैं, and they drop that drug right in the core of the tumor. They make sure that the drug should be there in the core of the tumor. So now what will happen? जहां जहां पे drug absorb करेगी, the drug will start like diffusing in the neighboring cells. जहां जहां तक ये drug diffuse करेगी, all cells will be killed indiscriminately. Because if a CTL came to this place, that CTL become an indiscriminate killer. If a CTL does not come to that place, that CTL will be normal. So there are two types of CTLs. The CTLs who are coming here, the CTL who are not coming here, who are like dealing with the rest of the body. Those who are dealing with the rest of the body, they will stay same, but they drop a small amount of drug right in the core of the tumor, that all the CTLs here will become indiscriminate killer, and they will start killing the cells here. So you will see the core of the tumor will become hollow. The cells will be dead, and they use it calculatively, so it should not harm the bad thing. So if you drop it right in the middle of the tumor, you can guess what will happen. You will confer more damage to the bad cells as compared to the normal cells. But if someone drop the same injection or the same drug right in the within the good cells will be killed as well. So it's require a very delicate surgery or incision, and what I must say, the transfer of the drug to the right place to avoid these two questions. ठीक है? Or you can design. The future will be that वो जो लोग कहते हैं ना, the people who used to speak कि भी एक चिप लग जाएगी वैक्सीन से, उससे हमारा ईमान बदल जाएगा, हम खुदा पे भरोसा करना छोड़ देंगे, and then दुनिया में जो है वो इस्लाम खतरे में पड़ जाएगा, something like that. There are rumors about like this. साइंस अभी यहाँ तक नहीं पहुंची है, लेकिन जो करीब तरीन जगह है ना साइंस की, जहां पर the nearest possible scientific endeavor जो आप कर सकते हैं, वो ये है कि आप एक CTL को डिजाइन करें. You can design a CTL, a cytotoxic T cells, in such a way that it can recognize its target. कि ये अपने target cell को recognize करता हो. You can design or engineer the CTL in this way. Let's suppose if that target cell is present within the body of किसकी body में डालें? काशिफ. It is in the body of the Kashif. First करते हैं, this target cell lives, or it is specific to the Kashif's system. So अगर ये CTL is like shared by all of us, it will not going to harm any of us, because that CTL is designed and engineered to kill a one particular cell which is Kashif specific, which is present in his body. So सबके body में अगर वो चला जाएगा through air or through anywhere else, everyone else will be nice and fine, and it is harmless to everyone else other than that one cell which is present within his body. It is called ये है target killing. यानी कि that the nearest possible things you can do, you can use biology to kill people specifically is this one. Please don't use it. I just shared this thing with educational purpose only. Okay, नीचे title लगा, it's only for educational purpose. Okay, so last, how it execute the function. You can see the combination should be stable and strong. That combination require the communication between the it's not your internet, it's my internet. Okay, let me take you to the slide so that we can conclude it. Almost done. मैंने अभी आप लोगों से पूछने, आप लोगों की बारी नहीं आ रही, सारी मेरी बारी आई जा रही है. बोलिए. जी जी.
एक क्वेश्चन.
हाँ, why not? Question से बड़ी.
Sir, even if we stop the PD1 receptor, we still have the CTLA4 receptor. So do we deal with both of them or only block one of them?
Yes. How we use the CTLA4, I have explained that thing earlier. They have created a drug, and as a student, you should chase the name of the drug as well. Okay. When I said they have created a drug, as a university professor, you are the one who should chase कि वो ड्रग का नाम क्या है, वो किसने बनाई, कौन सी कंपनी ने बनाई, कैसे पब्लिश हुई, उसका डाटा कैसे है. Okay. Please be like this. That will increase our efficiency. I will show you the way, the interesting directions, and you will like go and fly on those on skyways. So here we go. So as a stable connection is required in order
Sir,
to
I have a question.
जी रोहा शाबाश.
Sir, is PD receptor on T cell is responsible for this purpose only या फिर सर उसका भी और भी कोई purpose है?
This purpose only. Nature, when nature is creating the immune cells, I mean, think about that you are creating a policeman, and you are giving that policeman a gun. If this is a gun, I'm not a good drawing. If this is a gun, you are giving that policeman a very special thing, a gun, a license to kill. So you need to tell this policeman, you need to educate this policeman very well to use this gun only against the criminals. You educate them, the law and enforcement agencies, that what are the characteristics of the bad guy, when you are allowed to take your pistol out, when you are allowed to shoot the person, and when you are not allowed to shoot that person. So similarly, when the nature is creating the killer cells, the CTL cells, nature give them a receptor, PD1. Okay, nature give them a receptor to check whether the other cell belongs to us or not. Hope this answers the question.
Yes, sir. Thank you.
Okay, good. So once that connection is established, an established connection means you have like a direct communication between MSC and TCR along with the antigen, and then you have a supported co-stimulation. In case of CD8 cells, you have a supported co-stimulation by CD8 receptor as well as through integrins as well. Once that is established, the CD8 cells, they started releasing these granules. They have granules containing the bad thing. They started releasing those granules. If you see those granules, they're like small, like vacuoles or like lysosomes. They contain important chemicals, particularly enzymes. So those granules, they have like a cell membrane structure, lipid structure on their outer boundary, something like this. So those granules can easily penetrate the cell membrane. Further, they also release the perforins, which help the cell membranes and increase the pore size. So whatever they are releasing, whoever the cells present around them, those chemicals will be received by the neighboring cells. Once the neighboring cells receive those chemicals, they are proteases. They are active acids. Excuse me. Active acids and proteases. So they will become dead as a result of those things. And we have gone through this. It's a lethal hit, actually. So that is how they're able to kill the bad cell. One more way to kill the bad cell is this one. I have shared this thing as well, but just for your, I want to talk about this last thing. The B, we have studied a lot. Okay, perforin, granzymes, the enzymes actually kill the cells. Look at this one. I have just talked about this thing with you, but there is another way the CTLs has it, and it's a very dangerous way. I believe that this might be used for terrorism as well. I don't know. I'm not a villain. I don't think in this way, but as a researcher, I believe that this is the most easiest and quickest way to kill a cell using FAS-L and FAS receptor. FAS is the receptor. FAS-L means the ligand or the ligand. So this cell, the CTL, it interacts with the help of the FAS-L present on the surface with the FAS receptor on the cells. Almost all the cells we are carrying, it contains the FAS receptor. It's a death receptor. No one can touch it. It's a death receptor. If that receptor is activated, the FAS receptor is activated, it will trigger a cell signaling cascade, which leads to apoptosis, dying right from the inside of the cell. And that is an exact example of the kiss of death, touching just one receptor, triggering a cell signaling pathway, which eventually leads to apoptosis. And apoptosis is like a death right from the inside. And you will see the things which are really responsible for our own lives, those things are actually became the reason of this our own death as well. For example, I want to stop actually, I want you guys to speak, but just this is an interesting example. You guys understand the mitochondria much better than me. It contains all the mitochondrial matrix. It contains all the enzymes required to do what? The most wonderful thing out there, energy production, ATP synthesis. Glycolysis was done in the cytoplasm. The Krebs cycle and electron transport chain in the mitochondria produces ATP, and there is no life without ATP. There is no life without mitochondria. Now think, there is no life we know without this organelle, without the function of that thing. All the material present within this mitochondrial matrix. So in one of the way, not in this direction, in this way, I'm just trying to tell you that how cell always have a way to kill us, to kill itself. उसके पास कोई ना कोई तरीका होता है अपने आप को मारने का, हमेशा होता है उसके डिस्पोजल पे. So if an apoptotic pathway is activated, like through P53, I'm not going into the detail, but if the P53 got activated, it actually do a lot of things. One of the things, it stopped the cell cycle. You don't need to worry about this thing at this time. And the other thing is that it triggers the BCL pathway, and that changes the pore size of the double membrane of mitochondria. The mitochondrial membrane becomes porous. It releases its matrix, especially the cytochrome C, into the cytoplasm. Once the cytochrome C is there in the cytoplasm, our cytoplasm contains a lot of proteases. Most famously, they are known as caspases.
Caspase.
Shabash, thank you. So caspases. Like we also contains a lot of endonucleases as well, but those endonucleases are present in our cytoplasm to destroy any nuclear DNA, to destroy any DNA which comes to our cytoplasm. They're very, very bad, very dangerous, but they're not killing our own DNA. That's another story. Why? So the proteases and caspases present in our own system, they are not supposed to work because our cytoplasm contains a lot of important proteins. Cytoskeleton is there, protein structures are there, enzymes are there. So they kept inactivated. This cytochrome C can activate the caspases and convert them into an activated version. So what will happen? Those things become monster after that. You can say the caspases, and they start killing all the beautiful structures present in the cells, including the cytoskeleton, including the other enzymes, and the cell become dead right from inside. And this is called apoptosis. So this is another way of killing the cells. Okay, hope this thing is going to help you guys. This have changed your perspective about molecular immunology, what that exactly means when we use the term molecular. Molecular means studying about the phenomena at a molecular level, knowing about ourselves, that how we able to save ourselves from the disease at a molecular level. I believe that I will be very happy if you guys are different today than the start of your semester, and you have having more information about molecular immunology and about these things.